Okay, so as if you came in early, you'll note that I had mentioned I will hand the exam back in lab on Thursday. We had quite a range in scores, I'll say so, but for the most part, I think the average was like a 78 on the exam, so that was pretty good. But any specific questions I'll deal with on Thursday. So before the exam, we were finishing up, we had finished talking about the cell. Now, granted, that wasn't on the exam, but I just want to go over a little bit of review information for us. So let me move this thing off of the writing. Okay, so just as a quick review, prokaryotic versus eukaryotic cells. Notice the size difference between these two cells. That with the prokaryotic cell, and you noticed this in lab, I hope, on Thursday as well, when you were looking at your own cheek cells, and you could see the little speckles of bacteria on those cells, and the size differential between the eukaryotic and the prokaryotic cells. We're talking about 100 times size difference. And how simple that prokaryotic cell is comparatively. So being smaller, simpler in structure. What are we lacking on the inside in particular? What is missing in the prokaryotic cell? Yes. A nucleus? Yes. Membrane enclosed organelles. Thank you. So without having, as soon as we say lacking membrane-bound organelles, that wipes out everything except one kind of organelle, which is what? That all cells have? Ribosomes. Correct, ribosomes. So much simpler, but we also know that all cells have to have what in them? What information? DNA. Okay, so they do have DNA. It is just not enclosed in a membrane-bound region. Instead, we have what we call the nucleoid region. So it still does have DNA. Now, we will cover DNA in another couple lectures, and we will see that as opposed to having distinct, separate chromosomes, the chromosome in a bacterium is a circular unit of DNA. And so we see the size differential. In the eukaryotic, we see they're much larger cells, that they have more complex structure, and we see membrane-bound organelles and a nucleus. So there's many types of organelles in that cell. So what I'd like to do now, if this thing will go ahead and move for me, again, some things that we will see on prokaryotic cells that we may not see on eukaryotic cells, or on only a very few, will be flagella and cilia. And this is, again, just a cartoon prokaryotic cell. So it's showing all the possible things that it could have. Very rarely would they have both cilia and flagella, but we're trying to give you the real decked out model here with everything on it. So there can be prokaryotic flagella and cilia, and what would the main purpose of them be on a prokaryote? Movement, exactly. How do flagella move? Like propellers. And how do the cilia move? Like the oars on the Viking ship, exactly. So they can move in that fashion. Notice that, again, as we mentioned, with all cells, they are going to have DNA, ribosomes, and a plasma membrane. Animal cells. Animal cells, notice now we're at eukaryotic. So we begin to see the emergence of many different kinds of membrane-bound organelles. Now, what we're noticing in animal cells is what is lacking around the outside that we have in plant cells? Cell wall, exactly. So the outermost layer of the cell is the plasma membrane, although what can be on the outside of the plasma membrane? How come when you take a shower you don't fall apart, Marcus? Extracellular matrix, exactly. So we're going to go from here on to membranes, and we're going to see how important some of this extracellular matrix is going to be in holding that cell, cell-to-cell contact in animal cells. Now, obviously, that's not quite as important in protists that can live as independent eukaryotic cells, but when we get to animal, remember, what defines an animal? What are the characteristics? We're going way back now to one of the first lectures. What defines an animal? Well, they have mitochondria, but all eukaryotes do. But what differentiates animals from other members of the eukarya? Yes, Shanak. They have no chloroplasts, which says what about their mode of nutrition? They're chemoheterotrophs. And do you know how they get that nutrition, animals? How do they take in their nutrients? They eat. They eat. They eat their food. Okay? So animals eat their food. Okay? They're chemoheterotrophs. What else about animals? What makes them different than a protist? I know you guys know this. What makes them different than a protist? They're bigger, but what makes us bigger than a protist? Yes? We're multicellular. Thank you. All right, yeah, really. We're multicellular. We're multicellular chemoheterotrophs, okay, that eat their food. Because if I just said chemoheterotrophic multicellular organisms, there's another kingdom in the eukarya that would fit that. And what would it be? Fungi. Exactly. It would be fungi. But what's the difference between fungi and animals? How do fungi eat? Yes. They're decomposed, but how do they get their nutrients within their bodies then? They don't eat it. They absorb it. Exactly. They absorb it. Okay? You don't go up to that thing of French fries and put your hand on it. Send out like enzymes that decompose the French fries and then absorb the nutrients and all that wonderful, saturated and unsaturated fats and starches from those French fries through the palm of your hand. If you were a fungus, that's what you'd do. Okay, so thank goodness we're not doing that. It would take a very long time to eat those French fries. Okay, so this is just a single animal cell here. Okay, showing you just one. And this, again, is all decked out. It has all the possibilities of what might potentially be on an animal cell. There are not that many animal cells that have a flagella, except for one particular portion in the human life cycle. And what is it that has a flagellum? Sperm. Exactly. Do we ever see any cilia on human cells? Yes. Where? Yes, and a lot of the endothelial, the linings. Okay, so think of your respiratory tract. It's what helps get all the mucus out of your lungs, that sort of stuff. Okay, so now we see on the outside a plasma membrane. We go to the inside. We see what we know we're going to find in every cell: ribosomes, DNA. But where's the DNA now? In a nucleus. Okay, and within that nucleus is a nucleolus, and that is responsible for helping to make more of the organelles that all cells have, which is prokaryotic or eukaryotic. They have this one particular organelle that's not membrane bound. Ribosomes, okay, and then we're going to see mitochondria, members of the endomembrane system, the rough and the smooth ER, the mitochondria, are separate. They're not generally called part of the endomembrane system. Okay, so we see all this. So what I want to do is quickly show you a little video, a tour of the animal cell, and it's a little cheesy, but I think it's a good one, so let's just go with it here. Okay. Okay, no, that's not the one I want. I want the one that goes like this. Tour of an animal cell. I don't know how the volume is here, so hold your ears for a minute. We'll see how this works out.
If you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Okay, so that was just a little quick overview. We talked about all of these individual components of the cell previously. Okay, now let's go back to where we were, and we're going to go on and look at plant cells. So as we look at plant cells, what's the first thing we notice about them? Cell walls. So plants have cell walls. Now, it's not a living component, but it is secreted by the cell. And what's the primary biomolecule that makes up the cell wall? Cellulose. Okay, cellulose. Okay, so that's what makes up the cell wall. What's another thing that we notice that's distinctly different now about this cell versus the animal cell that we just saw?
The shape.
Okay, so it has a more defined shape, and that will vary from, depending on the anatomy of the plant, where in the plant you're taking the cell, what it's going to look like. But something internally to that cell, what do you notice that's different that you don't generally see in an animal cell? Chloroplasts. Dan mentioned chloroplasts. That's true. And what do chloroplasts do? Photosynthesis. That's correct. What else do we notice? I heard someone else say it. Yeah, Malik.
Central vacuole.
The central vacuole. Okay, and what is the purpose of the central vacuole? Storage. That's correct. And mainly storage of what kind of products? Yes. Water. And water-soluble products. Sometimes we will find that some cells in plants are modified to store maybe things like oils and sometimes things like starch granules. Think of your wonderful baked potato. That's what you're looking at there. Okay, so those are some big things that we notice. Cell walls, chloroplasts, and central vacuoles. So what differentiates plants from the rest of the eukaryotes? A couple of the things that you just mentioned. Chloroplasts, which means that, what is their mode of nutrition? Okay. But Shonak gave us this wonderful thing about chemoheterotrophs last time. So what are we going to call plants? Yeah, William. Pardon me? Photoautotrophs. Okay, so a characteristic of the plant kingdom is photoautotrophic organisms. Photo referring to what? Light. Okay, so they can feed themselves. They can transform solar energy into chemical energy that they can then use to power the processes of their metabolism. Okay. Also, what we note is another thing about them, the cell wall makes them unique within the eukaryotes, but it's particularly what that cell wall is made out of that differentiates them from a couple other members of the eukaryotic kingdom, particularly the fungi. I don't know if we covered this. Do you know what fungi cell walls are made of? I heard it. Chitin. Same thing that makes up the shells of what? Crabs and shrimp. Yes, crustaceans can have some of that secreted to the outside of their cells. So that will make a shell. And then something else that separates. What makes a euglena, which you saw in lab, which was green and photosynthetic, what separates a euglena from a plant? What separates a euglena from you? Why does it go in protist and not animal? Single-celled versus plants being multi-celled. So they're multi-celled, photoautotrophic organisms with cell walls made from cellulose. Okay. So those are plants. Now, we've pointed out the differences, but look at the similarities. What do we see in there that we see in animal cells? A nucleus. What else? Golgi. ER, ribosomes, plasma membrane, mitochondria. Thank you. Okay, again, one of the biggest misconceptions is that plants do not have mitochondria. They do. Okay, as I said, plants do not live simply to make sugar to feed animals. Okay, they transform solar energy into into the chemical form of sugar so that they can power their own metabolism. And the leftovers are what feeds us. Okay? So let's do a quick little tour of the plant cell.
I have a question for you.
Yes, Jeremy.
Why do plants make extra? Like, it's not just like what they need, but make more.
Well, they will store it,
Okay.
you know, so they're, well, let's just face it. It's almost like a teenager. If you put him on a couch with a video game and a bag of potato chips, he's going to eat all of them that are there, right? So, but no, they just make more. They're doing it for storage purposes and for their growth, okay, and for reproduction. And oftentimes we stop them before they get to that stage, okay? In fact, we've domesticated, most of the plants that we eat are domesticated. They're not wild plants. They're domesticated plants that we've specifically bred for that purpose. Okay, so come on. Okay.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. All plant cells have mitochondria, just like animal cells do. Sugars produced by photosynthesis are broken down and converted to ATP in mitochondria. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
Okay. All right. So, guess what time it is? Clicker time. So get out those clickers. Okay. And so here's the first question of the day. Lysosomes. Okay, so lysosomes are members of the endomembrane system. And they contain proteins that are what? Hydrolytic, able to help recycle worn out organelles, used in intracellular digestion, or all of the above. The proteins in lysosomes can be used for all of those things. So which of those is the correct answer? So I'm going to let this go until about 50 seconds, or 45. I'm going to let it go 45 seconds. I think we've got about 52 people in here. Let's see how many have their clickers with them. I counted 52, I thought. Oops, I guess I miscounted. Okay. Ready? I'm going to hit the stop button. Last warning. Going once, going twice. Stop. Okay. And the answer is?
D.
Yes, correct. So, E, there's no E up there. Oh, maybe. Okay. All righty. All right. So yes, it's D. So overwhelmingly, we see that that's the case. Okay. Those always, I can never figure that out. Okay. Next, which of the following structures is common, means shared, to both plant and animal cells? Chloroplasts, mitochondria, rough ER, both B and C. hold it. This is, how about, oh, maybe I should make an E. Should I make an E for the person that likes to press E and come up with some other answer? Aliens. Okay. All right, 55, that was it. So which are common to both plants and animals? No E's this time. All right, both mitochondria and rough ER are in both plant and animal cells. Are we going to find chloroplasts in animals?
No.
Only if they've recently eaten a salad. Okay. Okay. Which of these organelles is not correctly matched with its function? Okay. Oh, look, you've got an E this time for that person that likes to press E. Nucleus with information storage, ribosomes, polypeptide synthesis, smooth ER, lipid synthesis, lysosomes, intracellular digestion, or rough ER, carbohydrate synthesis. So which of these organelles is not correctly matched with its function? Hearing a lot of clicking back there. These little castanets. Okay? I'm only going to let this go to 1 minute. We've got 10 people that still need to vote. 3 that need to vote. 2. All right. Okay. We've got a little bit of disagreement here. Does the nucleus store information? Yes. What are polypeptides? Building blocks of proteins, okay? So this is chains of amino acids, okay? Right. So ribosomes do build proteins and polypeptides. Smooth ER. What are the functions of the smooth ER? Lipid synthesis, but also what? Detoxification, that's another huge one. Stores what? What do you get out of milk? What nutrient? Calcium. Stores calcium, okay. Lysosomes, we just went through that. They can do intracellular digestion. And then the rough ER does not make carbohydrates. All right, we're on to the next topic. One of my absolute favorites. Biomembranes. Okay, you guys hear me harp on certain topics all the time. Shape and function. How important shape is. Molecular shape. Changing the structure of a protein or changing the structure of a compound is going to change its function and whether it can be recognized. Another one of my favorites is membranes. Because if we can create a barrier, we can create a gradient. And if we can create a gradient, what can we do? Pardon me? We can control movement, and through that, what can we do? I heard it. Harness energy. I want you to think of membranes like dams. If we have water running down a hill, unencumbered, It's beautiful. We have beautiful waterfalls and we have streams and all that, and it's beautiful. But we don't have the ability to have that water work for us. On the other hand, if we construct dams and we control the movement of that water down that hill, we can generate electricity that powers all those wonderful games in Las Vegas. All the one-armed bandits. We run all the lights. We can do many things. We can even supply drinking water, obviously. But one of the major functions of dams is that we can harness kinetic energy, the movement of water, and transform that into electrical energy that we can then use to power any number of different items. So whenever you see an organelle that is filled with membranes, it gives you a pretty good hint on what that organelle is going to do. So think back to those two organelles, two of them that we just looked at, that are just filled with membranes. And what are they? Mitochondria and yes, chloroplasts. Okay. So chloroplasts are busy transforming solar energy into chemical energy, and the mitochondria are taking food energy and converting it into another chemical energy in the form of a particular molecule that you're going to learn to love called ATP. So we've got lots and lots of membranes associated with these. So let's look at biomembranes. They form boundaries in and around cells. We already looked at the plasma membrane. Now these membranes, these biological membranes are what we call selective, they have selective permeability, meaning that some substances can cross them more easily than others. And these biomembranes are made up of two of the big biomolecule groups that we covered, lipids, and in particular one kind of lipids. What kind was it? Phospholipids. And proteins. So, what kind of sandwich is it? Peanut butter with jelly beans. Peanut butter with jelly beans. Okay, I saw that a lot. Okay. So, we have our phospholipid bilayer. And in that phospholipid bilayer, I want you to notice that what's towards the middle? What are those little dangly things? Fatty acids. Okay, and what's to the outside? The glycerol and phosphate hooked to it, right? Instead of having triglycerides, having the glycerol hooked to 3 fatty acids, we have, instead of one of those fatty acids, we now have the phosphate. Okay? And remember that the fatty acids are polar or nonpolar? Nonpolar, therefore they are repel water, which means they are called hydrophobic. Okay? And so are the phosphate groups polar or nonpolar? Polar. So they're going to be what? Hydrophilic. Exactly. So what I want you to now notice, though, is that we do have proteins. And these proteins are looking like jelly beans in that peanut butter sandwich. And some of them, I want you to notice, go the whole way through. Some may only be on the upper surface or the lower surface, which would be to the exterior of the cell or to the interior of the cell. And with those proteins, remember that you can have, okay, so we have an amino group, right? And the amino group makes up the amino acid makes up the proteins, right? The primary structure. And then you have an amine group, carboxyl group, and then you have, what's that other group called? The R group, which is variable. And remember, those R groups can have a number of different characteristics. Some are polar, some are nonpolar, some are acidic, some are basic. Got different types of characters they can have. And so, depending on how the tertiary structure of that protein, how it folds, these different R groups can be exposed. And if there's a polar area, where do you think it's going to be in relationship to the protein? Towards the outside. And if it's nonpolar, to the inside. Okay. So a lot can determine on where how these proteins are embedded in these fluid mosaics. So just to recall, we said that the phospholipids are amphipathic, meaning that they have both hydrophobic and hydrophilic regions, which, if we were to take these phospholipids and just take some phospholipids and drop them into water, they're spontaneously going to form a membrane. Because what happens is the polar heads are attracted to the water, the hydrophobic tails of fatty acids are repelled, they're pushed together, and we get this formation of a bilayer, as it's called. Now, just so you're aware, these are not solid. In fact, you will find that fatty acids can move within, or the phospholipids can move within the layer. You can have lateral movement. And remember, we're just looking at something in 2-dimension that's actually 3-dimensional here. So they can move within the lipid bilayer. They can actually flip-flop. So we could have an instance like this here where these two are actually going to trade places. What was on the top is now going to be on the bottom, and vice versa. Now, that doesn't happen as often, but it can. Now remember that those fatty acid tails, we talked about fatty acids and saturated versus unsaturated. So which one of them are straight lines? Saturated. Okay. So unsaturated have what kind of bonds in them? Double bonds. Okay, and that can affect the overall shape of the fatty acid. And so you can get these kinks in the tails. And so depending on the more unsaturated fatty acids there are on that phospholipid, the more fluid the membrane. The more saturated fats, the more solid or viscous, thicker the membrane is. Also, very important in membranes, but not talked about all that often, are the different steroids, particularly cholesterol, is an important component of many membranes. And having them in there, embedded within these membranes, can also affect the membrane structure and affect its fluidity at different temperatures, how cold or heat tolerant it could be. It also allows us a place to potentially attack with pharmaceuticals. Remember, I've tried to mention this when I can, but I've actually in grad school took a fungal physiology course, and we were at the time working on a fungicide that one of the grad students ended up actually determining the mode of action, and it was an ergosterol biosynthesis inhibitor. And what it did was it was specific to fungi, and they could use it, and it would not allow the fungi to make the right kind of steroid to fit in the membranes, and the membranes of the fungus just fell apart, and it couldn't grow properly. And in fact, that's what fluconazole is, if anyone's ever had to use fluconazole. And this is many of the things that you put on for athlete's foot and that sort of stuff. It attacks those sterols in the lipid bilayer. Okay. Now, we said that they are selectively permeable, so some things are going to get through and some are not. And what you'll find is, what's the middle part of the layer? What's in the middle part of your sandwich? The peanut butter. Okay. But the peanut butter is what?
Fatty acids.
Fatty acids, okay? And so they're nonpolar. So some nonpolar substances can move through the membranes. Some small, now they have to be small, so some small hydrocarbons can get through. And also, small molecules may be able to move through. But larger and polar molecules cannot move through that membrane without going through some sort of a gate. So I'm just talking about being able to walk up to the membrane and just pass through without having to go through some sort of a gating system. So polar molecules are not going to be able to make it through the nonpolar fatty acid core of the membrane. So this is part of the reason why we have all of these proteins embedded in the membrane. And now they can do different things. These membranes, these proteins in the membranes can do different things. And they can help to determine the function of some of these membranes. Some of the membranes some of the proteins in the membranes are peripheral, meaning, what does periphery mean? If you're at the periphery of something, what does it mean? You're at the what? At the edge. If you're on the periphery, you're on the edge. So if we're on the periphery of Allentown. Okay. We're on the periphery of Bethlehem. We're on the outer edges of it. Okay. So peripheral proteins are going to be bound to the surface of the membranes. Integral proteins penetrate the whole way through the membrane. So they go the whole way through, from the hydrophilic phosphate groups through the fatty acid core and back out to the other side. So they're going to have different functions just based on this alone. Another thing that we'll find associated with membranes are carbohydrates. Now, these are not so much for gating, controlling what goes in and out of a membrane, as they may be for recognition and binding. And most of the carbohydrates that we find associated with membranes are actually what we call either glycolipids, so they're hooked, it's a glyco referring to the carbohydrate group to the sugar, to glucose to the glyco. Remember we talked about glycosidic bonds between carbohydrates. So if we've got a carbohydrate bonded to a lipid, it's a glycolipid. If it's hooked to a protein, we're going to call it a glycoprotein. So some of these are on there, and as we see, these are important, especially for animals, because this, again, keeps us from falling apart in the shower, okay, and washing ourselves away. It helps to keep the cells in contact, and it also helps in recognition. Surface features that can be recognized. So let's just take a second, give your hands a rest, okay? And look at this plasma membrane. And what do we see on this plasma membrane? Well, first off, I want you to notice that we have our typical lipid bilayer. Okay, so we've got, let's just go out here to the side so we don't get get too much going on the cell at one time. So we've got our phosphate glycerol groups to the outside, and then we have to the inside the two fatty acid tails. And notice that we've got some cholesterol, got some steroids, okay, which are also lipids, right? So they're down in there in the hydrophobic region. And some of these to some of these lipids, we have some chains of carbohydrates hooked. Now this is on the outside of the cell, so this is to the outside of the cell. So we've got that would be what's called a glycolipid, because it's a carbohydrate that's hooked to a lipid. We can also have carbohydrates that are just out on the surface here that are hooked to a protein. And so the carbohydrates that are hooked to a protein are called glycoproteins. We've got what are known as peripheral proteins. We're going to see how important these kinds of peripheral proteins can be when we start looking at aerobic cellular respiration. And things like electron transport. So these things must be doing something for on the inside of the cell. We've got integral proteins which can go the whole way through. So now we have a potential gate through which compounds can enter and exit the cell. And on the outside we have all of these extracellular matrix fibers. But what you're noticing is that they are attached to some of the glycoproteins. So these surface structures, these glycolipids, these glycoproteins, are extremely important in helping to organize the extracellular membrane. The extracellular matrix, of the cell, and helping to hold everything together and in its spot. Okay? So, do we have any questions on today's material? We're good? Okay, so I will see you guys on... Oh, and you've got your Into the Jungle readings in. Okay? And on the Into the Jungle assignments? Oh my gosh, that's going to take me... That's probably going to take me at least a week, because it'll take me a good 6-7 hours to grade those. And I don't think I'll have 6 or 7 hours to do that till the weekend. Pardon me? They were due at the beginning of class.